Hi everyone, I'm continuing with the gene mapping and the cloning of the disease gene applications. As we have seen the first application, the genetic markers are employed in the identification of the traits related to the disease or inherited abnormal conditions. These markers, with the help of these markers, the disease gene can be identified, abnormal inherited disease gene can be identified and it is rectified. And the genetic markers are also utilized in the identification of the species, individuals, and the organisms. With the help of these genetic markers, the hereditary gene can be identified based upon which the identification of the species, individual, or the organisms can be done. Thus, the DNA markers like the VNTR and the STRS are one of the first choice in the DNA fingerprinting, parental verification, and the maternal cell contamination. VNTR and the STR. These are nothing but the genetic markers which are used in the identification of the paternal verification and the maternal cell contamination. And the markers like the RAPD and the SSCP have the great value in the plant genetic studies such as the plant hybridization studies, DNA brocoding, and the plant breeding and the creating the stress resistant species. Genetic markers can also be used in the medicinal plant research and the phylogenetic analysis. In the phylogenetic analysis and the plant research, these genetic markers are used. It is frequently used in the marker-assisted breeding programs. In the breeding programs also, these genetic markers are used. Next, how to map a genome. And if we know the genome, how to map it can be learned. It is first done by the Alfred Struve created the first genetic map of a chromosome from the fruit fly, that is a Dorsala melanogaster, in 1913. He proposed that the recombinant frequency could help determine the locus of the gene. That is, with the help of the linkage mapping, that is unit of this linkage mapping is nothing but the recombinant frequency, the loci of the gene can be identified. The genes which are far apart in the chromosomes, they are likely to undergo the recombination separately. And the genes which are close to each other are likely to undergo recombination together. Which are far apart, they undergo recombination separately, and which are close to each other, they undergo the recombinations together. By finding out how often the recombination frequency of the gene, it is possible to estimate the distance between the genes, and this is known as the linkage map. By finding the recombination frequency between the two genes, the recombination frequency will be present. By finding out that frequency, it is possible to estimate the distance between the two genes, and this is nothing but the linkage map. The genes that are on the same chromosomes are said to be linked, and the distance between these genes is known as the linkage distance, which has led to the development of the first human genome map. And the two or more characteristics are inherited in the family, and the genes are together on the same chromosomes. If both the genes are together on the same chromosomes, then two or more characteristics are inherited in the family. In this, if we consider this, in which the two gene, that is the gene 1 and the gene 2, they are present on the same chromosomes and it is linked. Which are in this the gene 1 and the gene 2, and in this they are inherited. In this gene 1 and the gene 2, they are present on the different chromosomes and they are not linked. And here both are present on the same chromosomes but are at a sufficient larger distance. So they are not linked. And to produce the genetic mapper, the researchers collect the blood or the tissue samples from the members of the family, and some of whom have the certain disease or the characteristics. And the researchers then isolate the DNA from the samples taken from the each individuals and closely examine the sample. And one type of DNA marker called the microsatellite DNA is found throughout the genome and consisting of separate specific repeated sequence of the bases. The more DNA markers there are on the genetic mapper, one of them may be present close to the disease gene. And this DNA marker with the disease gene, the DNA marker associated with close to this disease gene is marked with and it is isolated and it is rectified. Next, As we have seen, there are two types of mapping. One is the physical mapping and the genetic mapping. And the physical mapping, that gives the estimation of the distance between the specific known as the DNA sequence on the chromosome. And the distance between these known DNA sequence on the chromosome, it is expressed in the number of base pairs between them. And the different types of technique which are used for the physical mapping are restriction mapping, that is a fingerprint mapping, and the fluorescent in situ hybridization, and somatic cell hybridization, and the sequence staggered site mapping. These are the different techniques of the physical mapping. Coming with the first, let us know about the somatic cell hybridization. This involves the fusion of the somatic cell growing in vitro, and the somatic cell hybrids can be produced between the two cells of the same species or the two cells of the different species. The two somatic cells, they get fused. That is nothing but the somatic cell hybridization. It is also used in case of the human hybrids. They are used for mapping of the human gene. And the radiation. Next is the radiation hybridization and the fluorescent in situ hybridization. It is in situ hybridization, and it is also called as the FISH. It is a powerful technique for detecting the RNA or the DNA sequence in the cells, tissues or the tumors. It provides the unique link among the studies of the cell biology, cytogenetics and the molecular genetics, and it is very precise and provides a spatial resolution of the morphological and the genomic structures. This technique is rapid and simple to implement and offers the great probe stability. The fluorescent indicators, and these are used for the identification of the, like the genetic markers, the fluorescent indicators, they are used for the identification of the respective gene. And the radiation hybridization is one of the technique where the it does not require the pedigrees of the family or the family member genotyping. Instead, in radiation hybridization, it depends on the somatic cell technique and on using the PCR-based probe to screen a number of cell lines that carry the various species of the human DNA. A monochromosomal hybrid or the whole human genome cell line can be used for building the radiation hybrid maps, in which the gene next to the gene the required DNA marker will be present, to which the radiation is passed for the identification of the particular loci. That is nothing but the radiation hybridization. Next, linkage mapping. One of the important subtype of the physical mapping is the linkage mapping. The genetic maps are also called as the linkage map, provides a rough approximation of the location of the genes related to the location of the other known genes. Relative to the other known genes, the location of the required gene is determined by linkage mapping. And these maps are based on the genetic function of the recombinant, hence the name gene mapping. If the recombinant frequency is between the two loci is 50%, then the loci are found on the different chromosomes or far apart on the same chromosomes. As we have seen in the this thing. If the linkage distance is more than 50% and it is present on the different gene. If the recombinant frequency is less than 50% and the loci are found on close to one another. They are close to one another on the same chromosome belonging to the same linkage group. For linked genes, the rate of recombination is proportional to the physical distance between the loci, and the distance on the genetic maps are measured in percent recombinant, that is centimorgans, or the map units, that is mu. And the linkage map, also known as the genetic map, it is a table for the species or the experimental population. It shows the position of its known genes or the genetic markers relative to each other in terms of recombinant frequency. If it is less than 50%, the genes are present on the different chromosomes. If it is more than 50%, the loci, the genes are far apart in the same chromosomes or present on the different chromosomes. If the recombination frequency is less than 50% and the loci are found close to one another on the same chromosomes, and the distance on the genetic maps are measured in percent recombinants or the centimorgan or the map units. Next, Human Genome Project. It is an international research by the US, that is the United States, and it is a 13-year effort monitored by the Department of the Energy and the National Institute of the Health. And it was started in 1990 and completed in 2003. The National Institute of the Health provides a $3 million for this Human Genome Project, and it is a the first step of this project was the construct of a map of the genome using the polymorphic markers or the genetic mapping, and these markers helps in identifying the location of the disease gene. The main purpose of this human genome project is the identification or the construct of the map of the genome using the polymorphic markers, the constructing of a map, and these markers helps in identifying the location of the disease gene. One of the priorities of the Human Genome Project is to map as many as genes as possible on the human genome, and they are mapped using the different types of mapping approach compared to the method used. Compared to the genetic mapping, there are various other methods are available, and using those methods, they are mapped. The genes are mapped on the different types of disease genes are mapped, and the portion of the cDNA, that is the genes are sequenced, and the sequence is used in the mapping of the gene, and these mapped products are called as the EST, that is nothing but the expressed sequence tag sites. The portion of the cDNA after the identification of the genes, the mapping of the genes, they are sequenced. That is nothing but the expressed sequence tag sites. And these expressed sequence tag sites, and they are not the polymorphics, but become the candidate of the possible disease gene once the location of the disease has been identified. After identification of the disease gene, these expressed sequence tag sites, they become the candidates of the possible disease gene. If we identify the express sequence tag sites, the disease gene can be identified. And what are the goals of this human genome project? To identify all the genes in the human DNA, and to develop the genetic linkage map of the human genome, and to know the functions of the gene, and to develop a technology for the management of the human genome genetic information, and to determine the sequence of 3 billion chemical base pairs that make up the human DNA, and to develop the tool for the data analysis to store all of these genes which are mapped with the help of data analysis, and to store this information in the public databases. And the future of human genome project. And the gene therapy can be a great field of application of the data found from the human genome project. And the sequence of the genome will be useful to treat any diseases. And the sequence, that is the express sequence tag sites of the genome, will be useful to treat the any diseases. Researchers and the scientists are now looking ahead to add the 47th chromosome into the humans, which would not affect the other 46 chromosome, but will stay inside the human cell to replace any gene that is mutated. And these are possible since the complete mapping of these identification of the 47 chromosomes and any other development which are possible after the mapping of the human genome is done, which is very difficult task. Next, coming to the second segment of the topic, that is the cloning of the disease gene. And what do you mean by clone? It can be anything identical. Here, in this case, it is the DNA or the gene. That is, making the copies or the clones of the DNA is nothing but the gene cloning. Another definition is that gene cloning is a technique combination of the restriction, digestion, ligation and the polymerization to make the copies of the DNA artificially, and this technique are widely used in the recombinant DNA experiments and the genetic engineering. Another definition is gene cloning is referred to as it is a process of copying the DNA or the gene through the recombinant DNA and the transformation and the gene isolation in order to get the copies of the gene from various downstream applications. And the cloning of the disease gene is done in order, and after the cloning it is variable number will be obtained and it is identified, and it is alteration is done on the disease gene so that it can be converted them into the normal gene, and the or disease is suppressed. Disease condition